Welcome back to the Classics of Immunology Journal Club. Before we get started, don't forget to like the video on the YouTube site and click on the bell to get notified for more, and subscribe and check out my website, kendallasmith.com. Click on the writing tab and you'll find the paper that we're going to talk about today, in addition to all the classic papers in immunology. And that's really the only way you can, if you're new to immunology, that you can really learn what's going on in this business. The paper we're going to review today is from Nature Immunology in November of 2007. And the authors are Pushpa Pandiyan, Lixin Zheng, Satoru Ishihara, Jennifer Reed, and Michael Lenardo. That's from the NIAID at the NIH. And the title is CD4 positive, CD25 positive, FOXP3 positive, regulatory T cells induce cytokine deprivation mediated apoptosis of effector CD4 positive T cells. So as I said, it was Nature Immunology of November of 2007. Now the background on this paper, and you have to really know the background, otherwise it's difficult to figure out why these people are doing these experiments. We first noticed cytokine deprivation induced cell death back in the earliest experiments that my lab was doing with CTLL, cytotoxic T lymphocyte lines that were growing in response to TCGF, which then became interleukin-2. And if you let the cells grow up to a reasonable density, and then you didn't replace the interleukin-2, and they would deplete the interleukin-2 in the medium very rapidly as the cell density got higher and higher. And in a very short period of time, less than 12 hours, they would just basically disintegrate, die. You look down at a microscope and there's nothing there, absolutely, because they would just pop, disintegrate. Cytokine, that came to be called cytokine deprivation-induced cell death, which is what this paper is about today. So that was 1977, 78. This is 2007. So 20 plus years later, the people involved in these experiments, Pushpa Pandiyan, was a postdoc in Mike Lenardo's lab at the NIAID, and that was in Bill Paul's department, the Laboratory of Immunology at the NIH. And of course, that was the Laboratory of Immunology was a big laboratory, and there were a lot of principal investigators working in the laboratory. And one of them was Ethan Shivak, and Ethan Shivak happened to be the editor of the Journal of Immunology back in the 90s, and he saw a paper coming across his desk as the editor of the Journal of Immunology from Shimon Sakaguchi, and the 1995 paper where he pinned down the ephemeral T cell that he had been looking for quite some time as to be a CD4 positive cell in the helper so-called subset. And it was interleukin-2 receptor positive, the alpha chain positive, which was also called CD25. When Ethan Shivak saw that paper, read that paper, he said, and I'm telling the story that he would tell at meetings when he was talking, and he was saying, this guy Sakaguchi is right, and he was one of the earliest proponents to take up the whole idea that there was and what he called, what Ethan Shivak called, a professional suppressor cell. And of course, back in the 70s, there was a phenomenon of T suppressor cells that had been championed by several individuals at that time. And that whole suppressor cell phenomenon, within about 10 years, in the early 80s, when the T cell receptor had been identified by Alice Reinhertz and then others, and when they cloned the gene for the antigen binding region of the T cell receptor, they couldn't find any suppressor T cell receptors, and that was the end of suppressor T cells. And so this is now, that was in the early 80s. Now this is in the 90s. And the suppressor cells have reared their ugly heads back again. So that they changed the name so that they didn't use because they knew that was not a good idea to call them suppressor cells, although that's what they did. But they called them regulatory T cells. And actually, I think I reviewed the paper where Sakaguchi was the one who coined that term. So this paper that we're going to talk about is about regulatory T cells. And Shivak went on after the 95 paper in the JI from Sakaguchi. The problem was that Sakaguchi was using in vivo assays to demonstrate the suppressive function of these cells, and they were difficult, and they really had to be a professional to be able to do these experiments. And Ethan Shivak thought to himself, what we all need is an in vitro assay for this, and so he devised an in vitro assay, and he published a paper in 1998 in the Journal of Experimental Medicine, which set the background for this paper that we're going to look at today. So it was almost 10 years before this 2007 paper that we're going to talk about today. And Shivak's in vitro assay simply was a T cell proliferation assay in vitro, whereby he varied the ratio of the so-called responder T cells that were trying to proliferate, and then the regulatory T cells that he would add in various concentrations to see if he could show that they would inhibit or suppress the proliferation of the responder T cells, which he was able to do. And in his experiments, he said that, if you take out the antigen presenting cells, the macrophages, the glass adherent cells, then you can't show this suppression anymore. So it was an APC dependent phenomenon that he was looking at. And he said, that he did experiments with what was called transwell, where a permeable membrane separated the two different populations, the responder cells from the so-called regulatory cells. And if you did that, then it wouldn't work anymore. And he, on the basis of that, he said two things. One is that it's not due to just soluble molecule interleukin-2 consumption, which is what everybody was telling you. Everybody at the laboratory, Bill Paul's Laboratory of Immunology, they said, it's just a cytokine sink. It's not, this whole phenomenon is not very important. And Ethan Shivak didn't like that because he wanted to make it important. So, so the transwell thing, but he interpreted the results of those experiments that he did as saying that they needed to come, the regulatory cells and the responder cells had to come into close contact. They had to touch one another. That was the background. And Pushpa Pandiyan, and I had her come to New York and give us a seminar and talk, and then afterwards about all of these experiments. And then I talked to her about what it was like there. And she said that the tenor of the laboratory of immunology, there were people on both sides of the street as to whether Ethan Shivak was correct in his interpretation of his experiments or whether he wasn't. And she decided to listen to all the stories, and she didn't have an extra grind necessarily. But she said, I don't think Shivak is correct. I think that what you're looking at primarily is the consumption of IL-2, which was normally driving the proliferative response of the responder cells. If you take the IL-2 away, they won't proliferate. At least that's what the dogma was. So she set herself out to do some experiments to really figure this out, to shed some light on this whole phenomenon. So in the introduction of this paper, she goes through several points. And the first point is that these regulatory T cells are supposed to suppress immune responses. But the regulatory T cells themselves, and this is another thing that Ethan Shivak had shown, is that they're anergic, meaning that if you activate the regulatory T cells through the T cell receptor, and you had to do that in order for them to be suppressive, but they wouldn't make interleukin-2, and so therefore they wouldn't proliferate by themselves. They wouldn't make their own T cell growth factor. You had to give it. You had to give it to them, and they required the interleukin-2 for their suppressive function. And the other thing that had been shown, in addition to the fact that that they didn't produce their interleukin-2, they could consume the interleukin-2 if you gave it to them and measured that whole phenomenon, and they would consume other cytokines as well. And this had all been worked out by various people in the decade between 1998 and 2007. And the mechanism, the molecular mechanism whereby the Tregs suppressed or regulated was controversial, as I just went through in terms of that. And there were all kinds of, at meetings, you go to a meeting and everybody was, it was like the Democrats and the Republicans today. The whole story was attacked by Pushpa Pandiyan. She looked at the phenomenon that you could overcome the suppression of the Tregs with interleukin-2. So that was part of the whole idea that IL-2 was just being consumed. And if you flooded the system with interleukin-2, then you could overcome this whole suppressive phenomenon. And in this paper, what she shows is, going back to the 1977, 78 days in my lab, she shows basically that what's really going on in this in vitro assay, as well as in vivo, which we'll get into, is that the regulatory T cells are binding and internalizing, degrading interleukin-2, and consequently causing the responder cells that are trying to proliferate in vitro as well as in vivo. They don't have any IL-2, and they undergo cytokine deprivation, apoptosis. They die. So going to the results in this paper, the thing about this paper is that you have to read it yourself, and I'll do the best I can to try to highlight what I think are the most important parts. But there are 8 total figures in this paper, and an average of about 5 subsets in each subfigures in each figure. So there's almost 40 figures in this paper, and each figure is the result of a different kind of an experiment with its own materials and methods and so forth. So you have to really study this. You can't just sort of read the abstract and move on. I think you should. This is worthy of a lot of effort on your part. So let's go to screen sharing. And this is figure 1 in this paper. And this is where she shows very nicely that regulatory T cells induce the responder T cells to undergo apoptosis in vitro. And in the A portion of this figure is a flow cytometry diagrams of conventional, the responder T cells in terms of their CD25 alpha chain, IL-2 receptor alpha chain expression on the ordinate here, and CD4 on the abscissa. And these are the conventional T cells. And after they've been purified, she's gotten 94% positive CD4 positive cells, only 1% IL-2 receptor alpha chain positive. Whereas the regulatory T cell compartment that she's purified, she's got 97% are CD4 positive, and 94% of the total are CD25 positive. And then in the lower panels here, what you see is the FOXP3 expression worked out through intracellular staining, and in the conventional, the T conventional is only 3% were FOXP3 positive, whereas 90% were FOXP3 positive. And then in the B portion of this figure, we're looking at the assay where we've got tritiated thymidine incorporation of the different populations of cells. These are the responder cells in the open bars, and then the responder cells plus the Tregs in the solid, and then responder cells plus control cells non-Tregs in the gray portion here. And so you can see that this is cultured 1, 2, 3 days, and these are treated thymidine incorporation with just the responder cells, nothing with the and so with responder cells altogether, you get thymidine incorporation. And then if you throw in the Tregs, it's markedly depressed, whereas the control cells, it's not depressive at all. This is activation with anti-CD3 and antigen-presenting cells, all of Ethan Shivak. This is anti-CD3 and anti-CD28 for the an alternative way without the antigen-presenting cells, but with the co-stimulatory molecule CD28. And here is really what I think is probably, they say a picture is worth 1,000 words. This is in vitro looking at the cells under the microscope with trypan blue, the vital dye that will be excluded from normal cells, but will be taken up by dead or dying cells. And so here we have just the responders. Here we have the responders and the control cells. And here we have the responders and the regulatory T cells. And so you see all of these blue cells here are dead and dying cells. That's really what you need to take away from this particular. In figure 2, one of the things that she has staining for apoptosis through staining, and you can see that she looks at this whole situation over time, and it takes. It doesn't happen right away. It's not an immediate phenomenon that happens within the first 24 hours, or even the first 48 hours. It doesn't go to peak, but it does so by 3 and 4 days. And so then the other thing is that she's looked at whether or not, if you flood the system, can you overcome the cell death, the apoptosis? With IL-2, the answer is yes. And then with IL-7, IL-15, IL-4, and IL-21, all those gamma chain, IL-2 receptor gamma chain positive cytokines will prevent the cell death, the apoptosis. That's figure 2. And I want to move on to the last part of this paper, to figure 7, because that happens in vitro. Now the question is, so what? Does that mean that that's the way everything is in vivo? And here is a classic experiment whereby others had worked out that the inflammatory bowel disease, if you set up a system where you can produce inflammatory bowel disease, and in this instance, this model system is that you put naive responder T cells into a skid mouse, and then you wait a week, and then treat the mice with regulatory T cells to see if you can suppress whatever happens, and then you wait 8 weeks later when they have a full-blown inflammatory bowel situation. And here you can see where they've just been treated with PBS, and here they are without any regulatory T cells, and there's massive infiltration of the mucosa, submucosa, with all kinds of inflammatory cells and so forth. But if you give those mice after only a week with regulatory T cells, you can markedly suppress that. This is the weight of individual mice that have been either given the inflammatory bowel disease in this instance, and you can see that the Tregs ameliorate that so that the mice are okay and they gain weight normally. And so that's really the bottom line of this paper, that Pushpa Pandiyan was able to show, and Mike Lenardo and the rest of them on this paper was able to show that Ethan Shivak was he may have been partially right, but he was not totally right, and that cytokine deprivation, apoptosis, was a big part of the whole phenomenon. So it stops screen sharing, and now move on to the discussion. Basically, they make the following points in the discussion. That point number one is that apoptosis is a major problem, a major mechanism whereby regulatory T cells suppress an ongoing immune response. They simply kill the responding T cells. And they do that by consuming the interleukin 2 that's being produced by those responding T cells. They in other experiments that I didn't go into, they looked to see whether or not they could have show evidence for active killing through things like tumor necrosis factor or perforin or granzymes, and they could not show evidence for that. So this was symptomatic and characteristic of cytokine deprivation apoptosis. That was then reinforced by the fact that if you could flood the system with interleukin 2 or interleukin 7 or 4 or 21 or whatever, you could overcome that whole cell death phenomenon. And they were also unable to find early on in the first 24, 48 hours evidence that the regulatory T cells had suppressed the production, the IL-2 gene expression and protein production of interleukin 2, which is what Shivak said that was really going on, and they said, no, it's not going on. The only thing that they didn't go attack experimentally was this Transwell experiment. They did the experiment and they found the same thing that Shivak had found, and that was that when you separated the regulatory T cells from the responder T cells, that you couldn't show suppression anymore. And Shivak had interpreted that by, as I said, by saying that, well, the cells, first of all, they weren't consuming any soluble molecule that could freely pass through the membrane. And second of all, they needed to have close contact with one another. They had to kiss one another. And what they point out is that the Transwell kinds of experiments separate the distance between the cells in the upper layer and in the lower layer in these Transwell apparatuses is a huge distance. And so it was very much way beyond the diameter of a single lymphocyte. And so that they doubted Shivak's interpretation. So that's how this paper ended up. And subsequently, as we'll get into, because we're going to dissect out the molecular mechanism of Treg suppression in the next few episodes of this journal club. And so stay tuned. And before you go, don't forget to like the video and click on the bell and subscribe and check out the website, the writing tab. And so thanks for coming. See you next week. If you're new to immunology, or you just want to delve into a certain aspect of this large field, and you want to get a grasp on things, you should buy my new book, Molecular Immunity, because it's a chronology of all of the important experiments and discoveries that have been made in the last 60 years. It's concise, but it's also very comprehensive. And it's the only such book that's been written since 1970. So you can buy it on Amazon, and together with the YouTube videos of the Classics in Immunology Journal Club, you can't go wrong.